All right. So, please introduce yourself.
Well, my name is Gus Kousoulas. I'm a professor of virology and biotechnology and head of the Department of Pathological Sciences at Louisiana State University, in veterinary medicine, as well as the Stanley Scott Cancer Center Medical School in New Orleans.
So, what did you talk about here at this conference?
So, I was very pleased in this conference for the Nanotechnology 2022, part of the nanomedicine sessions, to present some of our work on the use of viruses for cancer treatment. The talk focused specifically on how we use herpes simplex virus as a vector, not only for a therapeutic vaccine for melanoma and breast cancer, but even more importantly, as a prophylactic vaccine. We had proof of principle, a recent publication on this particular subject, that we could use this vaccine strategy to prophylactically protect against melanoma and breast cancer.
So, that would be amazing if you have a vaccine to protect against cancer. Could it be against all kinds of other cancers too?
Yeah, we already have examples of successful vaccines. For instance, against papilloma viruses, the Gardasil vaccine that has been used now extensively to prevent any cervical cancer for women that may be created 20, 30 years past their infection with papilloma viruses. There's also efforts right now for prostate cancer vaccine. Doesn't work as well as it should, but I think the principle of vaccinating against specific cancers, it's already proven, and it's just a matter of time that we find the right neoantigens. These are the changed proteins that could be used to successfully combat different types of cancer.
Does it have anything to do with what they've been doing with the mRNA? And is that nanotechnology?
So it is nanotechnology in essence. There are actually efforts to use mRNA vaccines for cancer purposes, for vaccines against cancer, as well as subunit vaccines, purified proteins. The difference between viral vector systems is that you could do multiple things, include multiple neoantigens, so you have a much better chance. You could also provide other inhibitors, whether it would be microRNAs or any other ways to shut down specific signaling pathways to really prevent cancer treatment.
Is it correct that you need to have something that's very immunizing to be able to vaccinate strongly against it?
Yeah, and this is correct. The current idea is to find neoantigens. These are really changed antigens, but also they need to be antigens that can generate a strong immune response that could eliminate certain tumor cells when they first are created. Obviously, with the advent of now molecular genetics, the ability for us to sequence the entire genome, we only have the genome, we have the transcriptome, we have the metabolome, the proteome. All this information together can be used to create vector vaccine approaches to combat cancer at multiple points, and I think that would be the ultimately successful. As we know right now, combination therapies and personalized medicines, personalized drug therapies towards cancer is actually a solution, and I think ultimately that's what will happen. The vaccine therapeutic approach would be one in combination with others, perhaps some anti-cancer drugs, chemotherapeutics that could be used in combination to create a much more efficacious strategy to combat cancer.
Last time we did a video, I think you mentioned the word adenovirus. Is that still in your thinking?
Yeah, so adenovirus right now is also used as a vector approach for cancer in an immunotherapy approach. This is one of the viruses. The difference with herpes viruses is that herpes viruses already have two successful products on the market right now. One is the T-VEC, which is the product produced by Amgen, which is really used to combat melanoma right now. And also another one, which is really another herpes simplex virus, G407, which now has been licensed for use for the treatment of glioma in children and adults in Japan, and it will be a matter of time that will be also licensed in the US and throughout the world. So we do have 2 successful herpes viruses to combat these very important cancers. Adenoviral studies have also going. There are also a number of other viruses that are being used right now in phase 1, phase 2 trials that to combat different types of cancers.
So it's 3 years ago around, we did the last video. So you have like the company that's been progressing and you're bringing, you say it's successful, so you're bringing this to market for sure, and how soon?
Well, the viral vector that we're using is being now tested as a vaccine against herpes viruses. That will be tested in England through a phase 2 combined clinical study. That particular study will actually pave the way for the use of the virus for other purposes, including cancer. And we hope that ultimately we'll move on to a phase 1 trial for the treatment of most likely breast cancer as soon as possible.
And with all the stuff that's been happening in the world the last 2-3 years, is it giving you an extra push and helping you get all this out?
Well, you know, cancer is one of the most important diseases. A lot of people suffer. There are different heterogeneous cancers of all types. And I think, you know, immunization, we have combated diseases like measles and mumps and polio, and of course papillomaviruses and you know, cancer, cervical cancer. We have combated coronaviruses using vaccines, so why not even cancer? And I think this is the era. A recent article in New York Times highlighted this fact that we're really in the era where cancer vaccines will be produced and hopefully will be very efficacious.
I don't know if it's correct to say that it's been hard to immunize with the coronavirus and all the what do you call it, the different variants and everything. And how do you think it's immunizing what you're working towards compared to the coronavirus, for example?
So the basic principle is that vaccines, some of them are extremely effective. They eliminate the virus or the cancer 100%. But most of these approaches are really actually synergistic to our immune system. So part of the idea and what's happening with current corona vaccines is not necessarily that they eliminate the coronavirus, but they reduce the viral load. And the viral load is at that level that then the immune system can actually successfully combat it. The same thing would be true for cancer vaccines. So what we hope is vaccines will be able to assist the immune system. And in fact, these vaccines are immunomodulatory, that is, they enhance the immune system. As we know now with cell cycle check inhibitors, with a lot of these boosting adjuvants that are used for cancer vaccines. So the idea is to really boost and modulate the immune system to successfully combat these cancers.
And how much do you combat the herpes, the problem people have with herpes, different kind of herpes? How much are you targeting to bring a solution to that?
So the vaccines for herpes right now that we are working on would be tested for both therapeutic and prophylactic. Therapeutic for those people already infected with herpes simplex type 2 and suffer a lot. This is actually the primary target of the company that pursues these rational vaccines, for which I'm consulting. And ultimately, that type of vaccine will also be used prophylactic in much younger ages to prevent the initial infection by herpes viruses. That will help a lot in the use of these viruses as a vectors for other vaccines, whether that would be malaria, influenza, corona, as well as oncolytic immunotherapeutics for various cancers.
As a final message, could you be optimistic in terms of maybe giving me a vision where there's no more herpes, there's no more cancer? Is it going to happen?
I think we are very optimistic, I would say highly optimistic, that we could really reduce herpes viruses to a very mild infection. And I think that we use the virus, herpes virus as a vector to successfully combat a number of important diseases, not only herpes, but also malaria, influenza, COVID-19, and other pathogens as they come through.
But when you use herpes as a vector, it's not a negative thing for the person who didn't have herpes before?
So these viruses have been engineered to be 100% safe and attenuated. They do not get into nerves. They do not cause any disease whatsoever. And in fact, the very fact that it's used right now for cancer treatment, that in humans, which we know that it will work.
All right. Thanks a lot.
Yep. Thanks.